scientists will come back to this location and do some hypothesis-driven research. I have a feeling the geologists will definitely want to be back here.
Sponge. Can we zoom in on that sponge when you have a chance, please?
Absolutely.
Aren't those two sponges?
Oh, yeah. Oh, I didn't even see the one in the middle. Okay, yes. So if we could...
Second.
It looks like that one sponge is right in front of a little pillow fragment.
This is also a really, really interesting sponge, at least as interesting as the one that we collected.
Is that again another hint, Chris?
No, I'd like a closer look at them, but they're two different ones. Superficially, I think the top parts of them look very similar, but as you can tell, the stalks are completely different. Perfect example between the different types of attachments.
It's interesting you say that, because I don't think the top parts look similar at all to the one we collected before. Or do you mean these two sponges with...
Before, I mean the two look similar to each other from this distance at least.
Oh, I see. My apologies, Chris.
You know you have joy lock on.
Thought it was off. Thank you. Yeah, video, if you want to comment, I'll try to follow you.
Hey, zooming in, and you're really quiet, Sean. Got you turned up all the way.
Sorry, let me...
I'm going to... I'll start zooming in.
Sorry, I adjusted my mic.
That's much better. Sometimes the mic gets turned around.
Yeah, sometimes I reposition it on my head just for comfort, and then I just kind of adjust the mic away from my mouth unintentionally.
So this is a sponge that I'm
Let me
pretty
know when you're
sure
ready
is
for a
in the
tighter
genus
zoom.
Saccocalyx,
Slightly
and it's also in the family Euplectellidae, and you can see the stalk is not fibrous at all, and it actually attaches via a plate to the substrate. Plus, if you look at the actual meaty part of the sponge, it's got a big osculum on top, and then it has all of these little side projections coming out of them with little oscula, or osculi, I guess. I don't know the plural of it, and that's pretty characteristic of the of that genus. So it's a really beautiful example of a sponge. We've got these in Hawaii, and they're fairly common. This looks at least pretty similar to the Hawaiian variety.
Ready for a tighter zoom?
Scott France just chimed in that he thinks he saw this thing or something very similar in the Aleutians too, so he thinks this group is pretty widespread. And then after this one, you'll see the difference with that other sponge. If we can pause for a moment and check that one out too, and you'll see it's going to be quite different in a number of respects.
Okay, video's happy. Ready to zoom out, pilot?
Ready on you.
Zooming out.
Thank you, video.
Also, just a little fun fact, it's probably taking the water in from the sides and then expelling it out that big, huge hole on the top.
Let's take a look
And
at this
some people have speculated that creates what's called a Venturi effect, where the current going over the top actually draws the water through the sponge and through its sides at a faster rate.
All right, video, I think I got it. If you want to come in.
Zooming in. Holding here.
Diva, what are those things?
That
at the
is
bottom?
just beautiful. Chris, do you want to explain?
I have no idea what I'm looking at here. This is maybe a hyalinide? I don't know.
The spicules are so beautiful.
Well, actually, is there any possibility of sampling? Because we would
All
have
right,
to
I've sent
stop
out an
soon.
email to Charlie Pomponi.
It's kind of like the other one, but you can see the bottom part.
I heard
is
you. Hold on. Give me a second.
completely different. Showing those spicules attaching to the substrate.
So, Chris, is there any chance we'd want to sample this, or do we want to continue?
Oh, I would love to sample it, but some of the other people might want to hold out for some other type since we got a sponge in the basket. But this is quite unique. But
Any of the scientists,
Copy that,
shoreside
Rich.
scientists, is there anything you'd prefer? We've only seen one coral so far for this entire dive,
Right, I might keep in mind that you haven't seen a lot of biology.
and we haven't seen a lot of biology at all. I mean, we may go upslope and find Utopia, but I doubt that very much.
And so you would collect this because it's like a novel thing that you aren't.
Do you want to go for a tighter zoom, pilot? Is
Yes.
that all
You
right?
haven't seen
So Shirley and Scott are saying sample it. Shirley and Chris
Hold
are
it.
saying sample it. Sorry, I'm clearly confusing people today.
1 sec, video.
Shirley and Chris are saying sample it. I haven't heard from any of the other scientists, but that's two people that are advocating the sponge.
the fact that Shirley and I really
A sponge taxonomist, yes, I did think that,
Yeah,
but
could I get a move actually
See,
the
this
other
will be
way
the
to
sponge
back
people
up?
dive.
Can you speak up a bit?
Alex
20
from Guam is
meters,
also saying
bearing
sample. So I think, I mean, the one thing you have to remember,
170
scientists, is that
at
if we do collect this
0.2
one, that is it for biology today. And we still have an hour and 15 minutes left
Good copy,
in our
Rich.
dive.
Do you think
No.
we should move forward a little bit,
Yeah.
or do you think sitting
Yes.
on these rocks?
No,
No.
2 hours and
Move
15
forward.
minutes left
Yeah.
in our dive. So
Copy that,
do you want to buy so early in the dive? Okay, everybody's agreeing to collect. So I think
So,
let's just go for it.
All right, copy that.
So
one. Just
yes,
set up.
pilots, we'd like to collect this one when you have a chance. Please.
So pilots, the ship had just stopped its
I
40-meter
assume
move
that,
to the north.
People who want to sample it
So
want to
then
make sure that those spicules
back
come
up
on board.
20
Is that
the exact
right?
same way. So,
So Chris, how crucial
timing's a little complicated.
Chris,
but
how crucial
I'm
is it
hoping
to
that
get
Sirius will end up,
the spicules at the
on
base?
top of you.
Okay. Should we move the ship back it off?
Well,
I
if
did
you
move
could
the ship
grab
back.
around the spicules and just pull it up, it should be just like a plant coming up
But
right up out of the soil. It
might-
should come up
It's
quite
tricky.
easily.
You might want to pause
Okay.
like 4 minutes before getting the craft out to make sure.
Okay. We'll do that.
Pilots
See,
and
Sirius is already
Those spicules just have little hooks on
kind of
the
starting to
ends, and
pass
they're
over.
kind of just hooking into the sediment and onto rocks and things, so it's not a real durable attachment. But
So you get any
even if
beauty
some of
shots
them
and get set up, but
broken,
we'll just be
it'll
ready.
still be okay.
Co-pilot, can you tilt down, keep an eye on him?
Okay, sure, Chris. Pilots, I'm not sure if you heard that, but our onshore scientist was saying that if you grab at the base of the sponge, so where the spicules kind of flay out, basically it should just lift off like a plant being uprooted.
Okay. Copy that. I'm just gonna take a minute to set up. And the ship, depending on where Sirius lands, we are gonna pause a few minutes to make sure Sirius doesn't
And
get
so Shirley
ahead.
Pomponi, one of our sponge taxonomists on the line, as well as the biology lead for leg 3 of this Marianas expedition,
So
is
we'll
saying
use
that
Sirius's
what is so
camera as the
exciting
judge.
about this sponge is that it looks extremely similar to the one which we were looking at just
That's it,
before,
yeah.
which is on a very
we'll
long
use Sirius's
stalk.
camera as the judge.
Whereas this one has
okay.
this very spiculated stalk. Well, it's not a
Sure, we
stalk really, just the sort of spiculated base which attaches directly to the seafloor. So,
That was-
yeah,
that
who
was like the
knows?
worst timing.
And they're pretty stumped by this, so it could
Call
be
for sample 'cause the
2
ship
new
had just
species of
stopped moving.
sponge in the ROV by the end of this dive.
Oh, it's close enough, right? Good pilot?
Yep.
All right. So we'll just wait a few minutes?
Hmm?
Okay. Well, actually, I don't know if you're aware, but we're going to pause a few minutes to make sure Sirius doesn't get too far ahead.
Sure, take your time.
Thank you.
So you can get yourself set up here and positioned.
So
So once you grab your controller,
if you come up like a plant, just get your fingers on the bottom.
well,
Slightly?
there's two options. You can come in from the side and you can try to grab the base of it, or you can try to come in from the side and cup it.
Okay.
Sounds like they want the base.
But they kind of want the base. So the other option is to come down like this and try to cup it like that.
I got the toy.
It's going to be difficult no matter what you do.
Yeah.
But you want to use a low setting on your grip force to where you can stop the jaws,
Okay.
because you're not going to want to fully close in on this.
Okay.
No, there's only one way to learn. So go for it.
All right. How does Sirius look?
I think if I could get the angle right, I would try to kind of grab the base with the tag on.
Just get it that low.
Yeah, but I don't know if I don't know if the geometry will quite work. But that would be, then you could just kind of grab it, yeah.
Yeah. Okay.
Because then you would definitely get the stock and you wouldn't damage the top.
Yeah. So we'll go for starboard box then?
Yeah, or port outer.
Or outer.
Or starboard.
We'd have to lift off again, correct?
Yeah, you're gonna have to lift
slow.
off again.
Okay, so we can do the same
I mean, you might be able to get it open, but probably not.
Yeah. Do you think that the previous sample would float? Out?
Do I? I would go starboard unless you don't feel comfortable going starboard.
Okay, starboard. Yeah. I think it looks.
I think
good.
we're good. I think we're at the end of the swing, and now Sirius should start backing up. I think we just caught it in time, so
Yeah, you can
feel
start
comfortable.
You can start positioning, and if we have to pull off, we'll pull off.
Yeah, can you come down a little
Yep.
bit?
So you want to set your grip force to
Super
something
light.
low that you can really control the speed on it.
I'm just gonna try one and see how that works.
I mean, you can also like stick your fingers into the soil too, you know.
Yeah.
But you want to be able to stop
Yeah.
and hold it. That's the key.
So this is one.
I mean, it might be too small, even the cup.
So just a note, it's very unlikely this is a brittle sponge. It's more going to be a spongy sponge. And so there's less concern about it breaking up when he grabs it.
Did you copy that, pilot?
I did. Yeah.
Good. I also have you on the utility cam here. You need it.
I see that. Thank you. So just touching. Yeah, I don't think I can get low enough where the blades would miss the bottom part. So I might just try to pluck it.
Sure.
Would you like me to zoom in a bit, or you have it here kind of where you are? Or?
I'm thinking about it, yeah.
Maybe if all those strands are really fibrous, just grabbing them might just not hurt them too much. Maybe
the fibers?
With the tips, we could ask watch lead, maybe.
I think you're in a pretty good
Sorry, what was that?
Watch lead. So I'm thinking about, I don't think I can get low enough where the blades would miss the bottom, so I'm thinking about just plucking it with the fingers of the craft. Do you think it will stay in one piece if I do that?
It's supposed to be spongy, so.
I was thinking the fibers on the bottom. Are they any tough? Are they any tougher than the rest? Or?
I have no idea. I mean, if
From what?
would it be possible to collect from the top down? So if you went from exactly above it and then sort of closed around it, would that work? I don't know.
That might just squish it.
So
yeah. Okay. This is why I'm not an RV pilot.
So Chris said it's spongy, and he also said that those hooks aren't going to be holding on too strong.
Yeah, I think if it's like a plant, the roots are usually stronger with the top.
He
But
said
I think if possible, we want to try and get a couple of the fragments on the bottom. However you think it's the best way of doing that.
Copy that.
So guys, this isn't going to fragment. This is going to be a soft sponge. So I don't think you have to worry about it. It's not going to be brittle. It'll just squish a bit when he grabs it.
But you don't think it'll tear, Chris?
Yeah, that's right.
Chris? Do you think it will tear?
only one way to find out.
It's very true for science.
Okay. I think just
Yeah, watch it. I'm gonna
go for it.
Yeah.
Go for it. Try to use your fingers. Like you got it.
Yeah, it's okay if the fingers, scrape the dirt as you go. Just get low.
Oh.
Yeah, you got it.
Yeah, nice.
Whoa. Looks like a lot came up there.
Okay. So
Those
bring
are the spicules
bring it
that
back
are
towards
hanging
you.
on to some sediment. So
I know.
Like the roots of
you
the
can
plant.
probably lock the jaw.
It is
You're
exactly
probably
like a
okay.
plant, isn't it?
It's
You've closed around
That's
it.
amazing.
Yeah.
So lift up the arm and bring it towards you so that you can move the vehicle.
Oh.
Maybe we can repot that in the pot with Lucian.
Yeah.
Looks like there's a few small pebbles in there, so you got some geology with it.
I'll make sure and throw those out, Chris.
That's pretty good. You're in a good spot there.
All right.
So you're safe to pull up and go into autos.
All right. Copy that.
Yeah.
You need to say something? Oh, keep it.
Just keep it there. Just fly. Yep.
All right.
Yep. And back out a little bit.
How's that look?
Yep, that looks pretty good.
Right.
All right, I'm going to open the drawer.
Copy that.
Are you doing starboard or port?
Starboard.
Yep.
Good.
Yep. All right, we're good.
Okay.
I wrote a note here for the next watch that the port sponge might float out. I'm not sure.
Oh, yeah. That's good.
All right, I'll keep an eye on you. Yeah, you got a bunch of spicules there. Where you going there, Sean?
Sorry, my shoulder got away from me.
Easy, easy, easy. There it goes.
Oh, it broke.
Yep.
The top came off.
So it was squishy.
Let's get the roots in.
Nice.
Sorry about that, those jaws.
Yep.
Opened up.
So that's the inboard box.
Inboard.
Starboard inward, yeah.
Thanks, Kapat.
That's fantastic. Thanks, guys. That's terrific. Great specimen.
Sorry I lost the top wash leads.
There were some
Such as life.
fragments in there.
Fragments are good.
Rotate the shoulder to port a little bit. Yeah, keep going. Yep, that's good.
Alright.
And shoulder up. Yeah, all the way. Yep, there you go. There you go.
Hydraulics off.
So either we're having a shift change among the ROV pilots or there's just a party going on in the front row. Either which way, I think we'll be here for a few minutes while everything gets straightened out after this fantastic sampling job that just occurred. So for those of us who may just be joining us on the line, we are here at Fina Nagu Caldera D, which is the oldest caldera among this chain that we're going to be able to image and sample at on this particular leg.
Good afternoon, gentlemen.
Tomorrow we will probably be going to Fina Nagu Caldera C, and then the next day we will go to the youngest, what we assume is the youngest in the chain, Fina Nagu Caldera A. We've been moving up a slope, and we are trying to hit a small local high that's about 200 meters higher than where we are now, and about...
You don't need the valve packs on anymore, do you?
I don't think so.
About
400 meters.
About 400 meters away. And so we've just been running up this ridgeline. We haven't seen a lot of biology today.
All right, guys.
But we have
got
seen a couple of species that look like they might be new.
we got
So we've collected
direction
two
of movement is going to be
sponges,
about 345.
Both of which, if they're
We've
not
got
new species,
almost
they may be
500
novel
meters to
occurrences
make
in this
before the end
area.
of the dive.
And then we've also collected two
And
rock samples, one at the beginning of our
so we've
dive,
gotta
one about
keep
halfway up.
Not a hurried pace,
so we
but a
can figure
consistent
out
pace.
the composition
to
and
get
perhaps
there.
the
As
age
of right now, you're directly underneath me.
this set of
Yeah.
volcanic
So-
rocks.
I'll let you get out ahead, and then I'll start a move, if that's okay.
And
Yeah,
right now
let
it
me
looks
get
like
out
we're
ahead.
Yep.
continuing our move upslope.
Consistently quick pace. Kill
Yeah.
lasers for me. I got
Yeah, I got you. Oh, yeah, you have a... Go ahead. And now it looks like I'm facing at 350 looking at the slope.
Okay,
Go ahead and get that ship moving.
copy that. 350 it is then. Bridge nav. 30 meters, 350 degrees, 0.2 knots, please. Correct, thank you.
And I think the high pack screen might actually be on camera 3, the stream, the third camera stream. And if you look at the high pack, which I believe is the bottom right, you can just see our ending waypoint right at the top. Oh, not anymore, you can't.
Yeah, I'm gonna have to.
Let me know if I start pulling on you.
Nah, you can't do anything about that. We're gonna have to wait for
Okay.
ship move.
I didn't want to get any lower delta than that.
So right now
That's
I think we're
not
just
the
waiting on a ship
that's not a
move before
problem
we
right
can
now.
continue
So
up
far
the
out.
slope.
Yeah.
Delta doesn't matter right now, really.
Okay.
Well, if you're going to wait on the ship move,
Give me a second, Kelly, I'm going to do the math.
Center, it's slightly in the upper panel. I think there's a very transparent, weird sponge that might be similar to the one that we collected the other day, that lyrate clatorizid.
Where's that, Watchley? I was shaking some.
Chris, where was that?
It's just to take a look if we were waiting.
And whereabouts, Chris?
Can't hear you, Watchley.
Whereabouts?
Kelly, it's going to be probably right around 1700.
Different Chris, sorry.
Whereabouts, Chris?
Apparently only in my head.
Okay, because I don't think anybody else in the control van saw that. There is something on the edge of this rock, if you'd like to look at that. Pilots, can we go look at that?
Sure. I think we can make it.
We just had a small communications mishap here in the van. We sort of may have switched a couple of headsets, which left Diva shouting randomly into the control room rather than actually using her headset for a moment.
And yes, Shirley, to answer your question, we did collect a Cladderized on dive 3,
I think so.
which was our deep dive down into Sirena Canyon. I think we caught it at about 5,000 meters depth.
That was right at the beginning of the
And
dive.
it was
5,000.
it was by far one of the best specimens I've ever seen in my entire life of anything.
This is that predatory sponge?
Yes, so
Zooming in.
it was, there. I can't remember what the genus is, but there's one that Imbari discovered a couple of years ago that's basically like a cross. So I think this is a stylastrid,
zoom.
Yeah, that's another hydrozoa.
which
is
we
that right?
no stylastrid, which is a type of coral.
Okay.
It's okay, close enough,
I'm
close
trying to
enough.
learn.
You're getting there. So anyway, so it's basically a couple of years ago, Imbari collected one that was sort of cross-shaped and had it basically like a fence, a cross-shaped fence. But the one that we collected was only one side of the fence. Basically, that was a terrible description.
Yeah, that
but,
really was.
yeah.
Sorry.
But it had 9 ophiuroids living on it commensally, and it was just absolutely beautiful. I will email you a picture.
Are we done with imaging? this
Yes,
one?
so sorry, pilot. We are good to go ahead. We've seen
Zoom
plenty
out.
of these for this dive.
Copy.
Full wide.
It was a bad description, but Shirley says she understood me perfectly.
Well, that's because she's a sponge person. You have to ask a geologist if they understood you. Or perhaps a physical oceanographer.
Yeah, when I'm that far out, co-pilot, I wouldn't get married to a delta number.
Okay.
It doesn't make sense. The delta's really for if I'm.
Yeah,
taking
you're
tight.
over 20. You're about 25 meters out from me.
Yeah. So as long as you've got eyes on me, we're okay.
All right, now we take the time to test which geologists have stayed on the line through the whole dive. So, Bob or Patty, anybody around want to tell us what we're looking at?
I'm just waiting until we stop looking at rocks, and I can tell you we're looking at a clump of probably very fine sediment.
No such luck, Patty. I think you're going to have to describe the rocks.
Okay. Now, these nice blocks of, some of them look as if they have
Thank you.
striated surfaces. Therefore, could be
Ship stopped. Do you guys want to go ahead and put in another one?
They're all
Looks pretty bare in here.
variable
Yeah,
sizes.
I think so.
This is obviously some sort of
snap zoom
a
on these
tailored
striations
or
down here.
free
Video.
Assuming striations.
and the fact that it has a dusting of sediment
Hold
on
it
it
here.
suggests that, it probably happened some time ago. There also
Let's do
seems to
another
be,
30 meters, same direction,
Zoom it out.
350.2.
a lot of the rocks have
Full wide. And Chris, can you check to see if your mic is turned around?
as well as
How's that?
That's much better. I think that's why Sean was so quiet.
biological growth that kind of wave around in the frost wash. But I really like the fragments that have sort of a triangular shape or a pop
Thank you.
with one curved surface opposite the apex pie. Those are typical of fragments from broken up pillow lava. And that's what a lot of these practice, like the one right in the center of the screen, moving down to the lower left now. That looks like a great pillow lava. The one we're coming to in the top center of the image has a lot of striations on it. That looks like a pillow.
Yeah, it absolutely has
And
lots
if
of
you
striations.
go over- Yeah, it looks like they have come from someplace further up slope that I hope we get to.
Yeah, so for people who are listening, one of the things that we are really trying to get to is that hopefully the local high will be the source of all of these displaced rocks. So we're hoping to try to find our outcrop. Thanks, Patty. That was really helpful.
We've still got approaching 400 meters to go, which we can make that if we continue at a not fast pace, but not dallying.
Okay.
You can get another ship moving.
All right. So science team,
Yeah, we already do.
Okay.
Diva just checked in with our navigator, and we're probably going to need to, I don't want to say make haste, but we're going to have to move along if we'd like to get to our last waypoint. So I think we're going to ask that we just keep moving, enjoy the imagery as it goes by, and unless we see something truly amazing, which of course it's all truly amazing, but sometimes you have to go where you're trying to go.
How much longer on bottom now?
We got, what time is it now?
Well, from a shore-based
We still
person,
got like 90 plus
Patty,
minutes.
I agree.
Yeah,
It'd be nice to get up as far as possible during the dive.
Yep, I think we're all in agreement now that we need to make a little run for it.
So previous watch said that they haven't seen anything steep or scary. It's all been this pretty much the entire way up.
Okay.
So
It's good to know. So watch leads, I know we're trying to make tracks, but I'm going as far as the ship, as far as I can. This is between me and Sirius right now, so if you see me kind of going back and forth, that's why.
Okay.
It's not that we're not still going uphill, it's just that I have a little bit of time to wait for the ship.
Okay. That one.
Trying to cover as much ground as we can.
Thanks. That'd be perfect. But we know the realities of you needing to move the ship and follow it, so we're good.
There's a Holothurian down there, yeah. Probably image plenty of these.
Is that a Holothurian in the lower center?
See, someone's excited.
That's good.
Yeah.
Yep.
Can we zoom in on him, please?
You want to go snap, Dan?
Sure. Zooming in. Holding here.
Copy.
Oh, that does look different.
So this Holothurian, to me at least, as a geologist,
partial
looks
wide.
very different
Zoom
than
in
some
out.
of the other ones we've seen.
Partial.
Yeah, it looks like it has little
Yeah,
ears.
thanks.
Little what?
Like it had little things at the front, like little. Yeah, we're all wiggling our
Okay.
fingers right now, trying to tell
Come on
Diva
in.
what we see.
Zooming in.
Does anybody,
Holding.
while Diva's looking it up in her amazing guide of animals,
Yeah, it's not in there.
so it's the little, not horns. What,
Yeah, yeah, I get
what
what
are
you
they called?
mean.
Antenna? What do you call that on a Holothurian?
I have no idea.
So, shore-based scientists, anybody?
First, I'm not seeing anything similar in
That's cool.
the guide.
Well, I think it's going to come out to be in a family Synallactidae. And again, I think this may be another type of Hansenothuria. That's a bad guess, but just from the skirt around it and the little projections on top. But I totally agree. We haven't seen this one yet.
I mean, it only
Nope.
has... Oh, he's going
I'm
to
gonna
swim.
zoom
No,
out
I think... Are you sure he's
as
on
he comes up.
Nepiastes?
Oh my God.
Because
Zoom out.
Synallactids don't really swim. I think he's in an Nepiastes, Chris.
Whatever it is, he's trying to get away from us.
Yeah, he doesn't... He's not happy. Yeah, and
Do you
plus he
want
doesn't have
lasers
that
on?
many projections on his body, just
Holding
the
here.
two
Okay.
sticking up
You're
by
at
his
partial.
head. I mean,
You
lots
can keep
of... It could
frame
be...
if you want, Dan.
There are lots of
Okay. We'll zoom in.
Holothurians that swim. Oh my god, amazing.
Copy, thank you.
Holding.
Let's do another
Look at
40
him
meters
swimming.
at 350.0.2.
I have mad hosiery in love right now.
Thank you.
Come in.
Zooming in.
Oh, the
Hold there.
extensions are
Holding.
called papillae? Is that right?
Papillae,
Yeah,
yeah.
so
There we go.
evidently calling
Nice
them little
dark
horns is
background.
probably not an appropriate term. I'll be quiet from now
So
on.
one of our echinoderm experts. Ooh, look at the ventral
Thank you.
side. Look at
What
that.
are the
so
dots
that's,
on it?
So you can see his mouth now, or maybe that's his anus.
Come in a little.
Zooming in a little.
Hold there.
Holding.
You can also see the 2 feet, the rows of 2 feet along the ventral surface along the bottom.
It's getting close.
Coming at me.
Yeah. It's kind of terrifying, actually.
That is actually. Come on.
Zooming out.
He's coming right at us.
Okay, so
Zooming
one
out.
of our echinoderm
Full wide.
experts at the University of
How
Guam
we
Marine
doing,
Lab,
co-pilot?
Alex
Looking
Kerr,
good. I'm trying to keep
has
you
said no, he has zero
12
idea
to-
what holothurian
well,
this is.
no,
He doesn't even know
about
what
15
family
meters
it's
out
in,
from
whether
me.
it's in Synallactidae or Late
but I'm
Magonidae.
losing you on camera.
The
You're going off to
extensions
my
on
port
it
side.
are called
Trying to
papillae,
follow me.
and this is definitely not Hansenothuria, he said, and it's definitely not Enipiastes, so
Like-
both of what Chris and I suggested
full wide
are
right now,
out
I
of the
can-
running. He's suggesting perhaps it's
hold it.
Benthigonie. Alison Miller's saying Paleopatides. I mean, why don't we just keep calling out holothurian names? We're bound to get it right at some point. But it is absolutely beautiful. Who wants to know that? I mean, they must to some extent. So, some of the people in our dry lab were asking whether holothurians have brains. I mean, they definitely have nervous systems.
I might have to let him go. He's
I'm at full wide right now. I can't
Oh my
focus any
God.
closer.
Oh God.
begging to be.
You're going to
So
be moving
Alex
underneath me, but I gave you
Kerr
a
from
20
the museum
Zelta.
has
So
also
I need-
seconded
I need to know when to
Alison
go.
Miller's guess
Okay.
and said
Yeah, we're gonna go.
he also thinks it may be
You're-
Paleopatides. So
Full wide, far zoom.
Copy.
that was great.
Far
That made my
focus, sorry.
dive. Yeah. Papillae.
I tried to give you a larger delta there so you could spend some time.
Yeah.
if you needed to.
So, instead of giving me those kind of numbers and things, I really just need to know when to go,
Okay.
so I can try to focus on the shot and everything. No, but you did everything perfectly. Just come up and then look down at me, you know.
Okay.
And that's when delta really matters, is when you're right beneath me, or when I'm right beneath you. So yeah, you can just follow me up now.
Okay.
Push way ahead.
Is Sirius zoomed out all the way?
Sirius is zoomed out all the way, yes.
Okay.
I have to say, I'm really learning a lot about biology on this trip. It's very exciting because, up until this point, I've mostly worked with hydrothermal vent geology, and so I understand the associated biology with that. But this is an entirely new set of things to learn about. Always exciting.
Yeah.
To answer the brain question,
Hey, pilot,
do
you want me to turn
holothurians
back
have
to the,
brains? No,
starboard?
they do not have brains,
bit?
Yeah.
according to
Okay.
Alex Kerr. But Alex, they must have some kind of nervous system.
Yeah, you come
just
back
not
online.
just not an actual brain.
We both need to come over, actually. 350.
Correct.
Okay. That's 350.
Copy. What was the brain determination? Brain, no brain?
No brain.
No brain.
No brain,
Central
but they do
nervous
have
system.
nervous systems.
Copy,
exactly.
no brain.
But I don't think that means that they should be discredited in any shape or form.
I think it was just a question from some of our physical scientists in the dry lab who are not biology people.
Right.
So they were just interested to know.
Can we maybe turn the...
Swing arms?
Yeah, we need to
Seems a little
adjust a
hot,
bit.
yeah.
It's hot in the front and real dark in the back.
We snap zoom on this real quick?
Sure.
What is that? Just a rock? Okay.
Holding. And zooming out.
Yeah, you come wide.
Okay,
Thanks.
coming wide.
Yeah, go for it, co-pilot. Whatever video needs.
Yeah, I'm not sure what your light orientation is now. It looks like the upper ones are out. Maybe the lower ones need to be tilted out.
Okay.
More.
Lower swing ons both out? Like deployed or
no, like
outward?
turned.
Okay.
Do you know if they tilted any when they were doing collections or no?
I'm not sure. That was before I hopped on.
I'll try those first. I'll try to go up with both uppers
Okay.
first.
Upwards with both uppers. Okay, they should be all the way up.
Okay. You need to come up with
Yeah,
me.
I'm on the way up right now.
Well, perhaps unsurprisingly for
And,
those people who
yeah,
have been on
350, I think.
this entire dive with us,
Oh,
we are
it's an auto
looking
heading.
still
It's
at a
kinda
scree or
swinging.
tailless
Yeah, I
slope.
got you.
Large areas of sediment
Did you want me
with
to try the
some
lower ones?
variably sized tailless blocks.
Yeah. If you're comfortable,
Can we go
I
snap
have
zoom
time.
here?
I can do it.
Video?
So just
go ahead, pilot. Sorry.
Can we come snap zoom?
Snap zoom.
What is that? Hold there.
Holding.
What is that?
Something
Besides
disturbed the
sediment.
I don't know.
That looks like there was something large and able to swim that took
Can you
off
come
from
wide?
that side.
Zoom in out.
Maybe a fish or a large crustacean or something.
Just look around here.
Did anybody see it?
Bruce, I think it was Munidopsis making an exit stage right.
Okay, thanks, Chris. I think I did see the crustacean. Couldn't figure out what it was.
looks like I have a slope to my port there.
Yeah.
Can we zoom on the coral or
Sure.
sponge stalk on top of the boulder, please?
Sure. What's your slant range to that? Figure it out center. To that face.
Can I adjust that?
Yeah.
Ship stopped, so you guys should be still got about 5 minutes, maybe 6 minutes of swing. But
Copy.
I'll hold us here till we figure out what that is.
Come on in, video.
Zooming in.
Oh, we want dark background, right? Yeah.
That'd be preferable, but
Yeah,
we'll work
sure.
with whatever we have.
No, you can just hold there.
Holding
So as
partial.
we were discussing before,
See if we
these
get-
rocks definitely have some kind of crust on them. Oh, that looks like a pillow back there.
Yeah, that'll look great.
They've got some
Yeah.
kind of crust. They're moderately sedimented.
And there's the toe.
So this was probably not a very fresh tailless slope. This probably happened a while ago.
It's looking like 15 degrees, but it looks like my heading's also getting turned.
Is it possible to zoom in anymore?
Yeah, we're just getting set up.
Sure.
Watching this.
Sorry.
No, no worries. Okay, come on in, video.
Zooming in. I'm going to go all the way in.
Copy.
Full zoom.
So those are not zoanthids. That is actually an octocoral. And isidid, according to Scott France, which are the bamboo corals.
Is this what we saw before, or is this just morphologically similar?
I'm not
I
sure.
think it's the same one that we saw before, and Scott wasn't, I think, participating at that point in the dive, so maybe he's seeing this one for the first time. It's got those very long spines on part of the polyp.
Yeah, I mean, I vaguely remember seeing this. Thanks, Chris. That was really helpful.
And if we could look at there's a sponge just to the left of it. If we
Okay,
could just
I'm
have
gonna
a
zoom
quick
out
zoom of
partial.
that, please.
Yeah.
And
It's on the other side.
it's on the other side.
Yeah.
I'll
maybe
come full
just-
wide.
Okay.
Let's test your pirouetting skills.
That's full wide.
That is full wide.
dig in there a little. Not that much. Okay, come. This isn't cooperating.
Thank you.
Alright, I made a mess, but it'll clear. Go ahead, video.
Zooming in.
I can try to get it from the other side if we think I'm
Holding
upstream.
here, partial.
Yeah, I'm just going to make it worse. Come on.
Okay, coming wide.
So I think this is a Caulophacus.
Full wide.
Yeah, thanks.
I'm getting above you.
Chris agrees as well.
Yeah, you're
It
stopped
does look
though,
much
right?
smaller and much younger
I believe
than
the ship
previous
move just stopped.
Caulophacus we've
Yeah.
seen.
I think.
So I think
you're about.
Yeah, Sirius should
Yeah.
be stopping
Okay.
in the next
I think that
60
was
to,
sufficient
if not already
anyway,
stopped,
pilot.
60 to 90 seconds.
Oh, we can. Okay,
So we can continue whenever you want.
copy that.
Do you guys
We
want
can
to
do a zoom here, Dan.
Sure.
That's what I was
Snap zoom.
looking for.
do you guys want to aim more towards that wall that Sirius is seeing and approach it perpendicular, or do you want to continue on
And
the general
holding
course?
here.
Copy.
I think that would be a good idea. But let's check with Chris there.
Yeah, just stand by, guys.
Copy.
And whenever you're clear, video will
Yep.
take off here.
Video's clear. Zooming out.
I'm gonna have to dust off here a little bit.
Full wide.
Copy. I'm gonna get out ahead.
See if you can go find that wall.
Yeah.
So we've got about an hour and a half left in our dive, and we are at 2,850 meters depth, approximately. And what we'd like to do in that hour and a half is continue upslope to see if we can find this sort of wall feature where we think a lot of this rubble and all these boulders have been moving downslope from. We
So-
have... Oh, go ahead.
I was just gonna say sorry about the mess. It's way more sedimented than I thought.
It's okay. We have 2 rock samples in our ROV, and we have 2 biology samples, both sponges, probably new species. So we've had a very successful dive so far.
What were we saying? A little to port, Brian?
Well, the general direction of travel is still 350, but we were seeing some harder returns, a little more to port on Sirius. So we were, but you should be out at them now.
Okay.
I think this is what we were seeing.
Yeah, I think this is what we
Yeah.
were seeing. So, what's your heading here? All right, let's do 340.
Yeah, let's do.
Okay, I'm going to go ahead and rotate to that.
Sounds good.
Bridge nav. 40 meters, 340 degrees, 0.2 knots, please.
How far was that ship move? 40.
40, yeah.
Let's see.
Thank you.
Andy, it looks like your
Looks like my swing arm.
lower
Yeah, I
starboard
see it.
swing arm is all the way down.
See it?
Sorry about that, Dan. That was a terrible sponge shot.
Yeah. It was a lot more
Okay.
sedimented than I thought. too.
Where do you need it for the better video?
Yeah, we definitely want to keep them at an upward angle since we're on a slope.
Uh-huh.
But so you'd want to like rotate the port light towards the right,
Port
and the
light.
starboard light towards the left, and that would. And I can't quite tell what angle they're at right now, but that would even out the light throughout the frame.
Hey, something was just to the left of that rock. If
Yeah,
you got
so
a
it was
second.
orange just to the left of the big boulder that we just passed. It's just gone off screen.
Kind of look like a dandelion, maybe.
Exactly what I was thinking, Brian.
So I have my starboard light all the way to
there
the
you go.
left,
I mean,
as
if we
is.
Can, it's right there.
Okay, that looks pretty good.
Okay, you want to try the port to the right now?
Yeah.
Okay.
Yep, that looks much better.
Better?
Yeah.
Okay.
Thank you. Standing by, pilot.
Come on in.
Zooming in.
So this looks like what we call
Holding here.
a dandelion.
Oh, cool.
And it is basically a benthic siphonophore. It's from the family Rhodalidae and possibly the genus Thermopalia. And we see them a lot in our deep
Try to
oceans.
zoom when you're ready.
But especially
Yeah,
around,
go ahead.
I've always
Going
seen
in.
them around hydrothermal vents. Maybe I'm just biased because I've worked quite a lot around hydrothermal vents. But
Holding here.
especially like, for instance, around the Galapagos, there are loads there. But yeah, so that you can see these sort of stringy like tentacles that they have attached, and that's holding them in place. So they're actually kind of suspended, but they, yeah. Well, no, I think they kind of stay for a while. Hemisessile. They can move, but I think they don't really like to.
Going in for tighter zoom.
They're just
That's
fascinating.
full zoom, actually.
That's my fault. So that's full zoom?
That's full zoom. Yeah.
Copy.
I think that's great.
Can we just zoom out a tiny bit to see what it's actually attached
Zooming
to?
out just a bit. Holding here.
You can turn on lasers too if we get a size. I think it's pretty small.
Lasers on.
Just beautiful.
It's hard to pick out. The sand is like the same color.
Yeah, zooming out a bit more.
Copy. Let's get. I'm gonna just tilt that for lasers.
Got it.
That's 4 centimeters. 3. Yeah.
We can zoom back in if you'd like.
I'm going to zoom in just for a few more seconds.
Yeah, go for it.
Then we'll be done. Hold here.
Copy. Holding.
Okay, that's great.
So
Zooming
basically
out.
this, what we're looking at is actually a colony of animals. Even though it may appear as one animal. It's many. All working together to do what needs to be done.
It'll be,
Much like us, scientists, ROV pilots, people on shore, people here, all
So it's
working
like
together to
Full
get what
It's
get
like an
done.
ant colony, basically. Like there are individuals which protect it. There are individuals which catch food. There are
lasers
individuals
back
which
off?
reproduce.
Yeah, sure. Thank you.
So yeah, that lighting looks pretty good, Andy. Thanks.
Okay.
It's just it'll be really important to remember when you stow them that you'll have to bring
Wait,
the
no,
port
that's
light
incorrect.
all the way left and the starboard light all the way
Hold
right
on.
before you stow them.
Okay. Okay.
So just remember to do that, and I'm sure Chris will walk you through
So
it.
Tim Shank has just chimed in and said that these, as I was saying, are typically found in hydrothermally active areas. So maybe if we do find some vents later on the cruise, we'll see some. But usually when the vent emissions are sort of waning or decreasing. So it's the most similar analogy for the dandelion is basically the Portuguese man-o-war. It's lots of different animals in one